Okay, hello and welcome everyone to this Integrated DNA Technologies webinar on Beyond Cloning, 101 Uses of Synthetic High-Fidelity Double-Stranded DNA. My name is Hans Packer and I will be serving as the moderator for today's presentation. The presentation today will be given by Dr. Adam Clore. Dr. Clore manages the Synthetic Biology Design and Support Group here at IDT, where he is also actively involved in product development, research, and biosecurity. Adam has his background in microbiology and biochemistry, and he earned his PhD in biology and systems science from Portland State University, where he studied the molecular genetics of hyperthermophilic archaea and their viruses. Adam's presentation today should last about 40 minutes, and following the presentation, Adam will answer your questions. As attendees, you've been muted, but we encourage you to ask questions at any time or make comments during the webinar, and you can do this by typing them into the questions box, which you will find in the GoToWebinar software at the right-hand side of your screen. And depending on whether you're on a PC or a Mac, it'll have a little plus sign or a little arrow that you can click and pop that out, make it bigger, type your question into it. At the end of the presentation, then I will direct the questions that we get to Adam, and we'll answer as many as we can. Also, in case you want to leave the presentation early today or you want to revisit this webinar later, pick up something you missed or get some additional details, we are recording the presentation and we'll make the recording available a few days after the presentation on our website, www.idtdna.com, and you will find that under our support tab in our video library. It will also be on our YouTube channel at www.youtube.com/idtdnabio. I also wanted to say hello today. We have a group of people at the Stour Institute who are watching this webinar, and I just wanted to say hello to them and to Kim Delventhal, who is arranging that with them. So hi, guys. So at this time, I'm going to turn over the presentation to Adam.
Thanks, Hans, and hello to everyone. Today I want to tell a story about a product that we really began thinking about a decade ago here at IDT. And the idea that we had behind this was to develop a product that was a double-stranded piece of DNA that was long enough to be used as a gene, but didn't require to be cloned into a plasmid or a vector or anything like that. The idea with this is that you could have more freedom to put this in whatever vector you wanted, and we wouldn't be constrained by biological systems in manufacturing this. Well, 2 years ago, this idea came to fruition in the form of a product we're calling gBlocks, and the full name is gBlocks Gene Fragments. These products are fragments of double-stranded linear DNA. We ship 200 nanograms. We normalize to 200 nanograms and ship that out dry, and typically we ship these in 2 to 4 business days. We've priced them at what we believe to be a pretty competitive price, and we designed these at the same time that Dan Gibson was perfecting the Gibson Assembly Method. And so these gBlocks were designed and tested using this assembly method. And I'll go into how this assembly method works here in a few slides. These gBlocks, though, are not limited to simply assembling in as genes and using the Gibson assembly method, but are really suitable for any purpose that requires double-stranded DNA of high fidelity and of lengths up to 750 base pair. And that's really what I want to talk about today. I want to go through some of the many uses that we have found for gBlocks, and more importantly, the uses that our customers have found for gBlocks. So before I jump into that, I'll just tell you a little bit about how we make gBlocks. We start with our ultramer oligonucleotides, and these are oligonucleotides that we make that are of the highest fidelity that we can possibly make. This allows us to make long oligonucleotides that are very accurate, and we use these as the base of assembly for gBlocks Gene Fragments. Once we assemble these with our oligos, we verify that we actually run these through a process that selects for the correct products and eliminates deletions and substitutions and truncated products. We then confirm that the sequence is correct using 3 independent confirmation methods. And once that confirmation has been done, we amplify, normalize, and ship these products out to the customer. And again, we're able to do this usually in 2 to 4 business days. I want to talk for a second about how we how we verify that these gBlocks are correct. This uses 3 independent processes, as I mentioned. The first is capillary electrophoresis, and we use this to verify that we have the correct length of product, and also that there is not additional products in there or byproducts in there that would be incorrect or truncated products. As you can see in some of the lanes here from the capillary electrophoresis run, you can see there's extra product in here. This allows us to remove this and remake these at the time of electrophoresis. After that, we use what I think is one of the most innovative QC measures, and that is the use of mass spectrometry. After years of development, we've been able to optimize our mass spectrometers to launch molecules that are sometimes in excess of 300,000 Daltons. And we're able to measure the mass of these molecules, not just to the degree of accuracy that tells us that there is the correct number of bases, but actually to the degree that tells us that we have the correct composition of bases. And so it's a very nice, very accurate way of measuring the purity and the correctness of the sample. Finally, once we're done with that, we also do Sanger sequencing just to verify that all of those bases that we know are present in the correct order. And so through these 3 QC measures, we're able to isolate gBlocks and only ship out gBlocks to people that are very accurate. The result is when we go ahead and clone these into a normal bacterial vector, we don't have to screen very many colonies to find the correct sequence. As you can see here, this is an example of cloning results from gBlocks ranging in size from 125 base pair all the way up to the 750 base pair mark. So you can see it's a very consistent percentage of gBlocks that turn up as completely wild type, that is completely correct inserts within the vector. And it's only in very rare cases that we see gBlocks that have less than 80% purity in there after cloning them. So it's something that is very robust and something that is very pure that we're able to ship out. Now, a lot of these, I should caution, a lot of these times when we see less than 80% purity in here, it has to do more with the biology of cloning these things than the actual gBlock itself. What we're able to see in many cases is that with certain sequences, there is toxicity or issues that the bacteria that we're cloning into doesn't like, and we get selection against the wild type product. As I mentioned, this gBlocks product is something that we launched 2 years ago, and that was in January of 2012. At that time, we launched a product that had a length limit of 500 base pairs. It had a turnaround time of up to a week, or actually up to 7 business days, so slightly over a week, and a price of $99. But since then, we've been able to increase the length of gBlocks that we're offering. We've been able to decrease the turnaround time now to 2 to 4 business days, and we've also been able to decrease the price. On top of that, we've also added mass spectrometry as an additional QC measure. One of the more exciting developments that we've had in 2013 was the launch of gBlocks libraries, and I want to talk about that in my next slides here. But before I do, I also just want to mention that we are not stopping this innovation, and in fact, February 10th of this year, we'll be launching our 1 kilobase gBlocks fragments. It'll be the same quality as our other fragments. You'll just be able to order longer ones. So let me jump into gBlocks libraries here. What gBlocks libraries are a gBlock that we allow mixed bases to be present in. This is the first iteration of this, and we're launching this fairly conservatively, at least on our website. So we have several restrictions on here. The first limits the mixed bases from being within 125 bases of either end. So the mixed bases need to be somewhat in the middle of the gBlock. We allow anywhere from 1 to 18 N or K mixed bases, and that can be a mixture of N's and K's. And just for anyone that isn't familiar with mixed bases, N represents a mixture of all 4 bases, and K is a 50-50 mixture of the G base and the T base. When we put these runs in, when you're ordering these on the web, the mixed bases need to be consecutive, so they all need to be next to each other. And the length of the gBlocks libraries that contain mixed bases is limited to 500 base pairs. So we can't go quite as long as the offering that we have for our standard gBlocks right now. We've beta tested this a bit, and then we've talked to customers about this a bit as well. And there's some common questions that I wanted to address. And the most common questions center around whether or not we can build more complex libraries, and that is libraries that have mixed bases other than N and K, multiple variable regions, or variable regions that are close to the beginning or the end of the gBlock within that 125 base pair area. We also occasionally get questions on specific codon substitutions. So an example of that may be a customer that would want a mixed base site that would only code for a couple of codons. Let's say maybe methionine, alanine, and threonine. So to answer these questions kind of in order, can we use mixed bases other than N or K? And currently the answer to that is no. And that has to do with the fact that we've worked very hard to get the ratios of mixtures right with N and K, and we're not confident that we've been able to do that with other mixtures yet. Can we have multiple variable regions? The answer to that is really maybe. We wouldn't allow that through direct online ordering, but if you have some sequences that you'd like to submit to us, you could submit those to libraries at idtdna.com, and that goes directly to my design group, and they can handle some of the things that we can't automatically accept online, and we can accept some gBlocks libraries with multiple variable regions in them. Again, kind of following up with that, it's not a hard limit on the 125 base pairs on the end. We can work around that with certain sequences, but again, those need manual reviews. And so I would encourage anyone that has questions about that to email those questions to libraries at idtdna.com. With specific codon substitutions, we don't have the technology that is robust enough currently to allow something like that to work. So that kind of sums up the questions about the complexity of libraries. If I haven't covered anything that you have a question about, please do email us at libraries@idtdna.com. A couple other questions that we get are why do we only allow 18 ends? And the reason for that really has to do with the complexity of the library. When we have 18 ends, it gives a total of nearly 70 billion possible combinations of sequences, which is nearly impossible to screen even with some of the most high throughput next-gen sequencers that are out there. It is also approaching the number of molecules that we're sending you in the 200 nanograms of material. And so we don't want to get to a point of where we can't represent the entire library in the number of molecules that we're sending out to the customers. The other questions are, can I get a discount, and are your libraries biased? We're not offering discounts right now. That may be something that's available in the future, but this is a very new product, and so we're not discounting it at this point. And with regards to how biased our libraries are, I have some data that I want to share with everyone. Of course, before I get to that, I'll just mention how we make and QC the libraries. The libraries are made in a way that's very similar to our standard gBlocks. And in fact, the non-variable regions are made as standard gBlocks and are QC'd as standard gBlocks. So they have the same quality as our standard gBlocks have. We then add a constant or a variable region into the constant regions. And we rely on a validated process, a process that we've spent years developing and validating through NGS to ensure that we have a very high representation of the DNA species present in the final material that we ship out. So this is an example of what some of our libraries look like. These are some of the validation libraries that we've tested. These are libraries that have a mixture of either 5 or 6 NNK molecules in them. And we wanted to look at the error rates. We first looked at the error rates of the constant region within these libraries. And what you can see here is that after next-gen sequencing and looking base by base at each position, there's a very low error rate. In fact, to actually see the error rate, we need to zoom into the 1/2 of 1%, the top 1/2 of 1% of the sequence to see. As you can see here, in most cases, more than 99.6% of the sequences are correct at any given base. So it's a very high fidelity. And in fact, this error rate really begins to rival the error rate of the machine, the sequencer that we actually use to sequence this. So we're very confident that we have a very high fidelity gBlock. But of course, what everyone is concerned about is the base distribution in the variable region. So if we look at a gBlock that contains one NNK codon, this is a typical example of what you would see. These are the reads by our next-gen sequencer for each base at each mixed base position. You can see in the graph here, it's a very even representation of the mixed bases across the board. And the K, the T and G mixture, is also quite even. A little bit higher representation of T, and I believe this is something that we have now adjusted for. But even so, it's within about 5% of the mark that we're shooting for, that 50% mark. Of course, when we look at more complex libraries, that was a big question. What would happen? Would this evenness continue? And the answer to that is yes. When we look at a library that contains 6 NNKs, we see a representation that is very even across all of the N's and very even across the K's. Again, a little more representation with T's in this particular run, but still a very even distribution of bases. So some examples of what people are using these for and what some of our beta testers have used these for is really probing things like binding site engineering, trying to modify binding sites of things like DNA binding proteins or catalytic sites of enzymes. We also have people that have used this for antibody engineering and optimization, and similarly for vaccine development and optimization. Several other, basically anything that you want to evolve or optimize a protein or a catalytic site for using gBlocks libraries works well. We've also had customers that were interested in systematic codon replacement, and that is a replacement of each individual codon across an entire coding region or catalytic site or whatever the site of interest is. And this is of particular interest to us and to our design group, and we can really, I think, help with this because what we can do is reuse some of the components of this as we build these libraries. And in doing so, we can decrease the turnaround time pretty dramatically and also provide a very significant cost savings to the customer because we're not building these individually. We can build these as more of a single unit. And then finally, we have some customers that are using multiple variable regions. And as I mentioned, in some cases, we can put multiple variable regions within a single gBlocks library. And in other cases, we can make 2 libraries that can be assembled through something like Gibson Assembly Method. Again, contact libraries at idtdna.com, and my group can help you work through any of these questions and design issues, and we can try and minimize the costs as much as possible. Finally, I want to mention the pricing and turnaround time of our gBlocks libraries. So our gBlocks libraries are priced the same as our 500 base, our 125 500 base gBlocks gene fragments as a base price. And then we tack on a fee for each mix base that is added into the sequence. So for the first mix base, we add $150 US, and if there's more than one mix base, we add $75 per mix base. Of course, these are US dollars. The prices vary slightly if you're outside of the US. And our turnaround time is 10 to 15 business days for these. So it is a more complex construction than our normal gBlock, so it does take a little bit longer. Okay. I want to go ahead and mention one last thing before I jump into the techniques here, and that is Rob Carlson's curve, or the Carlson curve. Rob Carlson is a very influential blogger and designer in the synthetic biology community, and one of the things that he's known for is trending the costs of reading and writing of DNA. That is, reading meaning the sequencing of DNA, and writing meaning the synthesis of DNA. What we've seen in the sequencing realm is a decrease in cost that is nearly exponential if we look at the per base cost over a period of years. And this has been likened in many times to Moore's law, at least in the way the curve is dropping exponentially. We haven't seen a similar drop in DNA writing, that is DNA synthesis. But if we consider gBlocks libraries in this, and the fact that in each gBlock library, you get thousands, millions, and sometimes billions of unique sequences in there, the cost of writing each of those sequences is dramatically lower than traditional synthesis costs, and in fact drives the writing cost of the Carlson curve down to a very low level. Okay. I want to mention also the biosecurity aspect of gBlocks and really all of the synthesis that we have here at IDT that involves double-stranded DNA. For anything that we make that is double-stranded DNA, we screen the sequences to make sure that we're not making something that is toxic, something that would harm our employees, and also not making sure that we're not making something that could be used in a way to harm individuals. When anything is flagged in our screens as toxic or as a controlled substance or from a controlled organism, it goes through a manual review that includes a review by myself and other members of the IDT team. So we're doing everything that we can to ensure that the sequences that we're making for people are safe and that they're getting in the hands of researchers who are doing the most good and staying out of the hands of people that would use them for means other than for good research. All right, I want to jump in now and talk about the uses of gBlocks and how researchers are using gBlocks gene fragments. And to start out with, I want to talk about gene construction and gene modification. When we think about gene construction, there's really three different ways that you can make genes. You can either start with oligos and construct the genes entirely on your own, which is the least expensive and most labor-intensive method of doing things. An intermediate method is to use gBlocks gene fragments, and in our minds, this has several advantages. The first is that the turnaround time is almost as quick as getting ultramer oligonucleotides. The benefit is you start out with a double-stranded fragment that is much longer than can be made as an ultramer, and that is already sequence verified, so you're already one step ahead of the game. Of course, we also offer gene synthesis for the person who wants to get it done as quickly as possible and get the genes in their hands as quickly as possible. Okay, so I want to talk about a couple of construction methods that we've seen. A group at Johns Hopkins University published this shortly after we launched gBlocks, and this was one of the first gene construction methods that utilized gBlocks gene fragments. What they were doing was looking at long exons and cis-acting elements within those exons that dictate the cleaving of introns and the processing of introns. So they wanted to go through and remove the cis-acting elements to create a negative control for these large exons. And rather than doing 18 different mutations using something like site-directed mutagenesis, they were able to just make this 1 kilobase exon using gBlocks gene fragments. They made this basically in an afternoon after receiving these. Were able to clone this in and proceed with their experiment. So it was a very nice way to use the gBlocks. Another nice example comes from the Church Lab at Harvard University. The Church Lab is interested in a great many things, but in one of their interests was looking at immune responses after flu vaccination and really trying to determine which antibodies bind the best to influenza. And this is something that is very important, especially with the influenza vaccine, because this is one of the vaccines that has relatively low efficacy compared to many of the other vaccines. So anything that we can do to improve the efficacy of this vaccine would be very useful. Their strategy here was to vaccinate patients normally and then wait a few weeks for the immune response to develop. And once that immune response does develop, they were able to take blood and using next-gen sequencing, go through and sequence and get a profile of the antibody response that was generated from this vaccination. They took the most highly expressed antibodies from this, the ones that are most likely to be from the vaccination, and isolated and analyzed the variable light and variable heavy domains of these antibodies. After doing that, they were able to construct the light chains and the heavy chains using gBlocks gene fragments. It works really nice for antibodies, particularly because they fall within the 500 base pair of our original gBlocks gene fragments lengths. So they were able to construct these, and because they had the light chains and heavy chains separate, they could recombine these and make a variation library of different antibodies. Once they expressed these, they were able then to determine the binding efficiency of these antibodies to the influenza virus, and they were able to find which antibodies work the best with the virus and use those as a real proxy to determine when they're getting good antibody responses from vaccinations. This allows them to optimize vaccinations and also optimize the vaccine itself to generate the strongest antibody response possible. So a very nice use of next-gen sequencing and a combination of gBlocks to build, essentially build a better vaccine. So the idea of making gene synthesis more affordable is something that we've thought about a lot with gBlocks, and I think a good example of that is if we consider a researcher that wants to build a 1200 base pair gene, and they want 8 different variations of that gene. Perhaps they want to vary the catalytic site near the center of that gene and look and test what these variations do. So if we decided to build this traditionally as a gene, the cost for this would be relatively expensive, even at 35 cents per base pair, which is a pretty good price for genes. Using 8 genes that are 1,200 base pair long, that would give us a price of a little over $3,300. However, this could be constructed with 10 gBlocks, where the variable site could be built into individual gBlocks that would be used, and the conserved site on the 3' and 5' ends could be made from one single gBlock. So this is 10 gBlocks at $89 per gBlock. That's $890, a significant savings over the cost of synthesizing these genes as a whole. Okay, I've mentioned the Gibson Assembly Method, and this is a method that's used by a lot of our customers, and actually a method that we use in-house for a lot of our gene synthesis. And for those that haven't heard of this before or not familiar with it, I wanted to describe it briefly. What the Gibson Assembly Method is a method that utilizes a small region of homology with each gene fragment to combine these and covalently bond these fragments together. And if you're doing this with a plasmid or with some other piece of DNA that can circularize something, in one tube you can actually construct an entire circular construct of multiple gBlocks gene fragments and a vector and put that directly into a cloning experiment. We can clone and you can move on and sequence. And this can all be done in a few hours, basically in an afternoon. So to jump into how this works, I'll zoom in on this a little bit here. The key to this Gibson Master Mix, which is, by the way, provided by New England Biolabs, if you guys would like to purchase it. The key to this are the 3 enzymes that are present in there. The first enzyme is a mesophilic exonuclease, and this exonuclease chews back from the 5' to the 3' direction on the DNA fragments. But because this is a mesophilic exonuclease, and because this experiment is ran at 50 degrees, it is rapidly degraded. So it only chews back a little ways before it falls apart, stops working. But it chews back far enough that it makes these regions of homology single-stranded. This allows them to hybridize to each other, and it allows the thermophilic polymerase and ligase to actually fill in these gaps and ligate and seal these gaps. And in doing so, you create a covalently bonded piece of DNA where these overlaps lie. So this is something that, as I mentioned, we use in-house, and many of our customers have used, and even with novice customers, people that have never done cloning and ligation before, this is something that works very robustly, and something that, frankly, I'm still just surprised at how robust this method works. Something that is really nice, and I would encourage anyone who's having trouble cloning to try this method. It's something that works really well. Okay, so we've talked a little bit about gene construction and modification, and now I'd like to jump into another topic, and that is genome modification. And I think in 2013, one of the most exciting advances that has came in the realm of genome modification was the utilization of CRISPR in genome modification. And for those of you not familiar with CRISPR, I'll take a minute to describe it. CRISPR stands for clusters of regularly interspersed short palindromic repeats. And these regularly interspersed repeats are found in a variety of prokaryotic organisms. In many archaea and in quite a few bacteria, these sequences are present. We've noticed these sequences all the way back from the late 80s on, and it was in the 90s that a researcher at NIH named Eugene Koonin postulated that this might be a prokaryotic defense mechanism against viruses. And it turned out that Eugene was right with this. We've discovered now that this is indeed a defense mechanism, and we've also discovered out how it works. It's basically a three-step process here. The first step is the introduction of foreign DNA into the CRISPR array. So what happens is the foreign DNA from a virus is injected into the prokaryote. The CRISPR-associated sequences produce proteins that are involved in the CRISPR the CRISPR cycle. And what a set of the Cas genes do, or the Cas proteins rather, is to cleave the double-stranded DNA into short fragments and insert that in between these between these repeats within the array, between the CRISPR array. Once these get incorporated into the CRISPR array, they're transcribed, and once transcribed, other Cas elements actually bind these RNA and use this RNA to screen DNA that's entering the cell and DNA that's present within the cell. And when they come across sequences that are complementary to the sequences that are expressed in this CRISPR array, they bind to that and cleave the DNA, cleave both strands of the DNA. So we get expression of that, and then we get neutralization through the cleavage of this. So this is all well and good for the prokaryotes, but it's even more interesting, I think, for us as researchers. And the reason for that is we can take these components and we can use them to target and create double-strand breaks in DNA. So to do so, we need three different components. We need the Cas9 gene from the CRISPR array. And this is the gene that actually holds everything and that scans along the DNA. There's also two pieces of RNA that are needed. There is the piece of RNA that actually holds the target, which is often called the CRISPR array, and there's also a trans-activating RNA. And this trans-activating RNA has a specific structure that binds and holds the target RNA in the Cas9 protein. Once this complex is formed, it can scan along, and when it finds a complementary sequence, it cleaves the DNA and creates a double-stranded break. Now, the one shortcut that researchers have found is that they can combine these two pieces of DNA into one piece of RNA, and this is often referred to as a single guide RNA, or sgRNA. This single guide RNA is around 125 bases long. So this is an example of what can be done with this. The real advantage to this is that this system isn't limited to just use in prokaryotic organisms. This can also be used in eukaryotes, including humans. So what we can do is we can express the Cas9 gene, and we can also express the RNA, the single guide RNA from this, and these two components bind together and create the double-stranded break. So we can use gBlocks for this, and a lot of our customers have done this. We've made hundreds and actually into the thousands of gBlocks for this right now. So a lot of our customers will use gBlocks to create the single guide RNA. And the nice thing about it is the most common promoter in eukaryotic organisms to drive this is the U6 promoter. And the combination of the U6 promoter and the sgRNA is around 450 bases long, so it's something that fits really nice in a single gBlock. We also have customers that have made their own Cas9 genes. This is particularly useful when you want to make a Cas9 gene that is codon optimized for a new organism or for an organism of your choice. So you can make both of these with gBlocks. Once you make these, you can combine those into an expression vector. There are expression vectors that express both the single guide RNA and the Cas9 gene on the same plasmid. These can be expressed on different plasmids to control them a little better. And in some cases, the U6 promoter with the sgRNA, the gBlock itself is just transfected directly into the cells. And by doing so, you can really modulate the expression of the RNA from this and really have a really limited expression. And this is important because, as with any genome targeting system, whether it be CRISPR's, TALENs, or zinc fingers, there is a certain degree of off-target effects. And so optimizing the amount of single guide RNA that you get into the cell so that you have enough to create the double-strand break at the targeted site, but not so much that you're creating off-target effects, is something that is really important to optimize for. Finally, we also have people that in vitro transcribe the single guide RNA and in some cases the Cas9 RNA, and then introduce those RNAs directly into the cell for expression. So once those are entered into the cell, we can look at what happens, and basically we get a double-strand break. And from that point, there's two things that can happen. The first, if there's no other DNA in there, is that particularly in eukaryotic cells, this will be repaired by a non-homologous end joining repair system. This is an error-prone repair system, and in the repair, it often produces indels, short insertions or deletions. And so by doing that, it often knocks out the function of a gene. So this is a good way to create knockouts of genes. But if you want to put an insert in there and not just knock out a gene, there's a method for that too as well, and it involves using homology-mediated recombination. So along with inserting the CRISPR elements, you can also insert a segment of DNA that has homology arms, and these arms will flank the double-stranded break site. And so when a double-stranded break is created, these homology arms can allow repair. And if there is a marker sequence or gene of interest between these homology arms, you can insert a wanted piece of DNA in the site of the double-stranded break. This was something that was first shown about a year ago by Daniel Dickinson and his colleagues. This is published in Nature Methods, and they were working in C. elegans here. So they use a Cas9 system to create a double-stranded break, and they also inserted a plasmid that had these homology arms. I believe in this particular paper they used homology arms that were around 600 base pair in length. So these homology arms efficiently recombined and inserted an element in the double-stranded break site. And so they were able to target not just a gene disruption, but actually insert a gene and a GFP element in there. And in using this, they used gBlocks to create the guide RNA. And so this was something that really helped them quickly generate these probes. Another example of the use of the CRISPR system is something that came out this year. This is published by Cheng and al. in Cell Research. And what he was able to show, or what he and his colleagues were able to show, was that you can use CRISPR not just as a method of creating double-strand breaks, but actually using it as a homing device. So what they were able to do here is show that you can deactivate the nuclease domains of the CRISPR, specifically of the Cas9 gene. And using this nuclease-dead mutant of the Cas9 gene, they were able to attach a promoter fusion to it. So this Cas9 gene still localized to the area where the target sequence was. But instead of cutting the DNA there, it localized the promoter to that area and promoted genes within that area. And they're able to show that they could affect efficient gene expression that was driven by this Cas9 fusion protein. So a very nice example of how to use CRISPR in a method that is not doing genome modification. Okay, before we move on to the next topic, I just want to mention this is just a small list of the papers that have been published last year in 2013 that used gBlocks gene fragments to make CRISPR elements and to use those CRISPR elements for genome modification. This is not an inclusive list, and this certainly does not include the list of all of the CRISPR papers that are out there. That is a very large list of papers. If you'd like to see a list of these publications and links to the PubMed sites for these, we have these listed on our gBlocks page. and that can be found at idtdna.com. Okay, so we've talked about genome modification. I now want to mention a topic that we never thought gBlocks would be used for, or we never envisioned that they would be used for, and that is using them as controls for qPCR and for SNP detection. So the first example of this was something that was done by Zymo Research, and they were able to, they were nice enough to share some of their early work with us, and we presented that in an article in our quarterly newsletter called Decoded. And that newsletter is available online for free. There's no login required for that. You can simply go to our website and read it if you'd like. So what they were interested in doing was measuring rates of methylation. And the way they were measuring this was using an assay that involved using methylation-sensitive restriction endonucleases. These endonucleases cleave DNA that's unmethylated, and they do not cleave DNA that's methylated. And so by digesting DNA with these methylation-sensitive restriction enzymes, they were able to cleave the non-methylated DNA. They could then use this DNA as template for real-time PCR and measure the shift in amplification curves that occur. And of course, the more DNA that's chewed up, the larger the shift is going to occur with the qPCR. But what they were interested in using gBlocks for with this was using a control that was completely unmethylated. And since our gBlocks are completely synthetically made and don't use any bacterial systems that would incorporate methylases, we're able to provide a product that we know is completely unmethylated. And so they use this as a non-methylated control for their experiment. It's something that worked very well for them. One of the other methods that several of our customers have used is using gBlocks to create a template for real-time PCR, and particularly for multiplex PCR. This is particularly useful for multiplex PCR because you can actually put multiple templates on a single gBlock, and by doing so, you always know that your controls are going to have an equimolar ratio of your templates. So you always know that you have the same number of each template in your tube that you're using. So it works, it makes a very nice control, and it makes it very easy to get relative quantification and efficiencies when you're doing multiplex PCR. Some examples of how this has worked in our hands is shown here. What you can see is that there's a very nice, very even representation of amplification from different templates that are located on the same gBlock. And you can also see that it's a very robust amplification, very repeatable, and something that produces very nice curves. One of the other very interesting techniques that we've seen people use this for is people that want to look at using a control that they can differentiate from the endogenous sequences that are present. And this is particularly useful when you're doing very sensitive qPCR. And particularly when you're doing very sensitive qPCR, you want to make sure that you're not contaminating your test samples with your controls. And of course, when you generate synthetic controls, you generate a large number of copies of the material. So it's very easy to get a couple of those copies to contaminate something. What the researchers here were able to show is that you can digest, that you can create a template that is slightly shorter than the endogenous template. They were able to create this template using gBlocks, validate that this template amplifies with the same efficiency as the full-length endogenous template, and then use this template as the control. Now by doing so, they have a way of distinguishing the control template from the endogenous template, and that's using a melt curve. What you can see in the melt curve here is that the endogenous template melts at a slightly higher temperature than the -10 base gBlock fragment. And by doing so, by using this melt curve, you can always tell that your template is either a control template that's synthetically made or an endogenous template. So it's a really nice control to run. Okay. Lastly, I just want to mention really briefly a couple of new technologies that are up and coming for that utilize gBlocks gene fragments. of course, our libraries have just came out, and so we have a lot of people that are trying those out, and we're seeing a lot of exciting new developments with that. Many of our customers are using our gBlocks libraries to create gene variant libraries and to create things like promoter variant libraries. And these really allow them to test the sequence space of these genes and these promoters and really dig down deeply into them and figure out how to optimize those and make the best genes and best promoters that they can. Another interesting method I just wanted to mention real quickly came out of the labs of Novartis, and that is using a technique that they're calling obligare. What this is essentially using a CRISPR system, or really using any genome targeting system. It could be CRISPR, it could be Talon, it could be zinc fingers, and targeting and inserting DNA into cells that lack homologous recombination. So they're using the non-homologous end joining pathway, but they're inserting an element of DNA in there. And the way they do this is to simply transfect in a high number of fragments of DNA, so that when this double-strand break is created and the non-homologous end joining method basically grabs ends and begins gluing them back together, that there's enough DNA there's enough inserted DNA in there that it actually gets inserted in between the break. And so by doing this, you end up getting an insertion of DNA in there. Now, you can't control the direction of the insertion, so it can go in either direction, but you can get a nice insertion of DNA. And this is particularly useful in cells that lack homologous recombination, or at least downregulated in homologous recombination. So some exciting new methods out there, and I'm sure in 2014 we're going to see a lot more methods like this. Okay, with that, I just want to go ahead and wrap up. I want to quickly mention our partners. We partner with New England Biolabs, and as I mentioned, they produce the Gibson Assembly Master Mix Kit, and they provide a very nice, very robust kit, and I encourage anyone who wants to try the Gibson Assembly to use the Gibson Assembly Master Mix Kit from New England Biolabs. It's a quick and easy way to get it done. I also want to mention synthetic genomics. We have recently partnered with Synthetic Genomics, and we're partnering with them in gene synthesis. They specialize in making very complex and very long genes up to 2 million bases in length. And because we're partnering with them, we can now offer genes up to 2 megabases in length. So I encourage anyone that has large and complex genes to give us a try, and we'll do our best to make a nice long gene for you. So with that, I think we can go ahead and end, and I'll hand this back over to Hans, and if there's any questions, I'd be happy to answer them.
Okay, I want to thank Adam for that presentation, and as Adam said, we can take some questions at this point. If you haven't already done so, there is a questions box at the right-hand side of your screen in the GoToWebinar software, and you can click the plus sign or the little arrow, depending on whether you're on a Mac or a PC, pop that out, type in your question. You can basically ask whatever you want to ask about gBlocks gene fragments. It's a great opportunity since Dr. Clore is basically one of our most senior support people. So we do have a couple of questions, and I guess I will get started with that. One person was asking what sort of design services that we offer for the CRISPR products.
Sure. Yeah, we can help in a variety of different methods with CRISPR. One of the challenges with CRISPR is the fact that this is such a new technology. You know, it really just kind of gained prominence in 2013. So it's relatively new, and there's a variety of different methods that people are utilizing to get CRISPRs into cells. And so one of the things we need to know from the customer is what method they want to use. Now, do they want to use a certain plasmid that would express both the RNA and the Cas9 protein? Do they want to do those on separate plasmids? we can help assist customers with that, and we can direct them to repositories where they can find these plasmids and where they can purchase them. AdGene, for one, has a very large repository of plasmids to do this in a variety of different organisms. So we can help customers with that. We can help them do some of the design. We can also point them to some design tools that are available online if they want to do it themselves. But essentially, getting some input from the customer, we can help them all the way from the beginning in choosing which plasmids they want to use, all the way up through the actual design of the target sequence, and if they need codon optimization of the Cas9 gene for whatever organism they're going into. So there's a great deal of information we can help them with.
Okay, and here's a great follow-up question to that. Is there additional charge if they ask for help when designing their gBlocks?
So we do have a design fee that is a $40 per hour design fee, and that comes into play after the first 2 hours of design. So we're happy to design for the first 2 hours for free. We're happy to give you that design work all the way up to the 2 hour time point for free. And afterwards, we can discuss what fees would be present. But honestly, in many cases, and in almost all cases, we can get all or almost all of the design work done within that 2 hour time frame, with the exception of some very large projects.
Okay. There are a couple of questions here about restrictions with manufacture of gBlocks. So one person, more of a comment that there seemed to be a lot of restrictions for gBlock synthesis, and then another person was asking if we had made any progress with making gBlocks fragments that are higher in GC content, and it would be helpful to know when was the last time they attempted to order, since some things I know have changed.
Yes. The rules is a project that's something that's very close to me. It's something I've worked on a lot over the past 2 years, and a lot of our customers that ordered at least 6 months or longer ago probably ran into a lot of restrictions with things. And I certainly won't say that we don't have any restrictions now, but we have a lot fewer restrictions. And the biggest change that I made to the rules came in late November of last year, where we were able to relax several of the rules, including rules about GC content, and particularly rules about hairpins and other types of repeat. We do still have problems with high GC content and low GC content, and sequences that are above around 70% GC content are often rejected. Unfortunately, we still do struggle a lot to manufacture those. Excuse me, sequences that are below somewhere around 22% GC content, we also really struggle with, and we have problems manufacturing. We also have problems with repeats, and so if the density of repeats becomes very high in certain areas, that can also limit our ability to make gBlocks. But one of the more interesting things that I've found in developing these rules and in looking at past manufacturing failures and successes was that oftentimes, and most often, our gBlocks don't fail for a single reason. They fail because of a combination of multiple reasons. And so we've designed our rules to reflect that. Most of our rules add a certain score to a total score. And it's only when that total score goes over a threshold that we reject a sequence. So it's often the case that it's more than one minor issue that causes a failure with things. One of the things that my design group really works on is working with customers that have had sequences that are rejected, and we can work through those. And oftentimes, if we can make just a few changes, we can increase the ability to manufacture these, and we can get those through the process. I will also mention that we're working on a major manufacturing change that we'll be releasing sometime this year that is going to greatly aid our ability to manufacture things. That's probably going to be incrementally released, and I think that'll come out partially in the first quarter of this year and the remainder in the second quarter of this year.
So if they do go through the website and they run into an issue with a particular sequence being rejected, at what point should they contact the support group? Should they contact you
I, you
guys
know,
immediately?
I think for some of our customers, they can look at, we do provide information about why individual sequences are rejected. For instance, we'll indicate where repeat regions are or regions of high GC contents that are particularly problematic for us. And so the customer can take that, and if they have flexibility in sequence, in changing their sequence, they can look through that and they can make changes themselves. But at any point in time that they're getting a rejection and they're just kind of stuck and don't know where to go, or just want some advice, I would suggest that they email genes@idtdna.com, and one of my staff can assist in that optimization or in making a small change. And again, oftentimes we can make very small changes that have quite dramatic effects on our ability to manufacture things.
Okay, here's and this is actually the last question I have on the list right now is, can I use the Gibson assembly method to assemble a highly repetitive sequence? And then they say they wanted to synthesize a construct in which 40 base pairs are exactly the same as the last 40 base pairs. So it looks like it's a 40 base pair thing that repeats.
Okay. The answer to that, I would say, is maybe. Part of it depends on how the Gibson assembly method is designed, and we can certainly help design things in a way that are going to maximize the chance of it working. Certainly, the customer could order that as multiple gBlocks, and as long as those had unique regions where the overlaps are going to lie for Gibson assembly, they could assemble that. And there's a fairly good chance of success. We may be able to make some sequences that would have 40 base repeats if it was just a single set of repeats. So one sequence repeated twice within a gBlock. We could look at those individually. The determination for that would probably fall on the exact nature of the sequence, the GC content of it, and the surrounding sequences. But we could definitely take a look at that. The next question for that would be, is it going to be stable in bacterial cells if indeed they need to be transformed into bacterial cells? And that tends to be a big stumbling block for a lot of people, and frankly for us as well when we're making genes. Bacteria likes to recombine things, and particularly if things are in high copy plasmids, the chances of recombination is quite high, and so these tend to be fairly unstable. Now, there's some tricks that we could use. There's some certain cell lines that tend to work better, and there's some low expression plasmids, if they're going the bacterial route, that could help minimize the chance of recombination with something like this, if they do indeed need to express this in bacteria.
Okay, looks like we got one more question here, which is, how much does it cost for the synthesis of a 2 kb gene in IDT? I guess that's going to depend on where they are. And do you clone into a specific vector which the customer provides?
Yeah. So in pretty much all cases, we don't clone into specific vectors. We just clone into our in-house vectors. Our in-house vectors are very generic vectors. They are, in most cases, puck-based vectors that we have stripped down and made as simple as possible. And we make that as basically something that can then be subcloned into any other vector. We try and do this to keep costs down and to keep our turnaround time quick, and also to minimize the amount of failures. It takes us a lot of time to optimize vectors into our manufacturing process that come from the outside, and so we've pretty much stopped doing that, stopped accepting vectors. When we're talking about the cost of these things too, it's very, especially something like a 2 kilobase gene, it's very dependent on the complexity that's within the sequence. And I would hesitate to just throw out a number for that. If you look on our website, our base cost for sequencing a 2 kilobase fragment is about 45 cents per base pair. Now, we do have some flexibility in that price, and we do adjust it accordingly based on the complexity of sequence that's available.
Okay, and with that, we're basically all the way through the questions and we're about out of time. So I want to thank Adam again for the presentation and for answering questions for us. And I want to thank everybody else for attending today and for providing those questions. I hope this was of value to you. This is one of a series of webinars that IDT will be doing on synthetic biology topics, as well as other molecular biology webinars. And those can be found on our website at www.idtdna.com, and you'll find them under the support tab in our video library. We also have a YouTube channel, which is www.youtube.com/idtdnabio. And the slides, we will be posting those at our SlideShare site, which is www.slideshare.net/idtdna. Those should be posted within the next couple of days, certainly by early next week. And at this point, again, just thanks for attending and best of success in your research. If you have any questions, just feel free to contact IDT. Thank you. Bye.
Thanks, everyone. Bye.